topics are the characteristics of organisms, the name and function of the body systems, and naming the cavities. Well, the characteristics of organisms, that's right, it's in a list on the first part of that first chapter. They reproduce, they respond to the environment. I don't know the exact characteristics that list entirely. But there's just a list, and so you'll just have to match that characteristic to some example, right? For example, one cell, in humans, one cell eventually develops into 3 trillion cells, which make up the body, right? So that's growth, obviously. All right, now the body systems, there are 11, and so I told you early on you need to make a little matching thing, the name of the body system and the function, okay? So, you guys can go through that. I might have examples here on the test. And then the cavities, dorsal and ventral. The ventral cavity has several subcavities, right? Pleural cavities, mediastinum, abdominal pelvic cavity, those sorts of things. All right, I think it's time to move into some of these diagrams, and we'll see some examples here. Oh, yes! Should I start over? No, yeah, I was only checking. Okay, so this is the actual figure off of your lecture exam right here. And it will be in grayscale. They don't publish color pictures for me for my test. And I notice here that you're going to have 2 sets of questions. One set of questions you'll answer with the letters. The next set of questions you answer with the numbers. Okay? So that means your Scantron, which you will bring that day, can either be letters or numbers. My machines, they read them anyway. It's just the position of the pencil mark, right? So you'll just have to, if you get the numbered Scantrons, then you'll have to convert the letters into numbers, right? A is 1, B is 2. You'll get used to that. It's a way for me to fit more diagrams on the test. Plus also, out of all these components, you can eliminate half of them for each set of questions, right? So it's a little helpful for you as well. So this would be typical questions. This cellular component is constructed of hydrophilic heads and hydrophobic tails. And in this one here, we're only using letters. So what letter would that be?
A.
So what are we talking about, hydrophilic heads and hydrophobic tails? The membrane, right? So here's the plasma membrane right here. That answer would be E. Okay? So far, some of you got one right. Now it says, remember to use the numbers. This organelle without ribosomes helps with cellular detoxification. What is the name of that organelle?
C. Lysosome.
Yes, a smooth ER or endoplasmic reticulum. Well, where is that?
Number 5.
Yeah, it's right over in here. It's number 5, no ribosomes.
Oh, just kidding.
Okay? Now, I never did tell you the names of either of those organelles, right? Okay, so you're going to have this kind of more flexible knowledge of how that works. All right, so the name and function for the cell, we need to know the name and function of the plasma lemma structures. I think we have another diagram in the test about that. Let's see. As a matter of fact, here it is already labeled. This is not an example of how I might label it. This is how it's labeled in your test. Okay. Don't get complacent. Don't get smug, right? And definitely don't spend 5 minutes identifying those components and call it good to go, right? Okay. Here's an example. We call this the phospholipid bilayer, right? And A is representing those phosphate heads right there, and B, the lipid tails, okay? Which one is hydrophobic?
B. B. The tails.
The tails are phobic, right? So you definitely need to know those vocabulary words. The tails fear water, right? The heads like water. Okay? They're hydrophilic. They love water. Okay? And that's how the whole membrane was organized, because those heads were attracted to the watery environment and the tails were trapped inside. Okay, so that's good. That's basic knowledge about the membrane. What kind of materials go through that membrane?
Ions.
Lipid-soluble materials can go through the membrane, like cholesterol and some hormones. If it was water-soluble, then there would be no separation of the watery environments on either side, right? So it's the hydrophobic tails that create the selectively permeable membrane, right? They're excluding anything that's water or water-soluble. Okay, so now we've already jumped from just naming parts to understanding how these parts function. Okay, and those I've left those in your notes there. Okay, so A and B are about the plasma lemma. C is a channel here, right? If you can't move things by diffusion right through the membrane, then we're going to have to start thinking of moving things through channels. Right. D here is talking about these structures that are sticking up off the surface of the cell. And that's called the glycocalyx. That was a word I didn't really put into your vocabulary. And actually the whole cell is covered with these antenna-like structures. What's the general purpose for those? Receptors in general, that's what they do. They make some contact with the outside environment, and then it changes activities on the inside environment. Okay? And D here is what? E.
Microtubule.
They're collagen fibers, they're the cytoskeleton, peripheral proteins, all of those work there. And so those peripheral proteins are, you've got to get going with that. The structure, moving, transport things around. They'll have to look into, again, what the cytoskeleton's basic function. So I'm not just going to say, name the components which represent the cytoskeleton, right? I'll give you a function, probably, or an example, and you go, oh, that's carried on by E, the cytoskeleton, all right? Got to look deeper. This is a concepts kind of knowledge, whereas lab is just memorization for the most part. All right, so much for the plasma lemma. Okay, your organelles, that would be, I would ask you questions that in the cell model that I asked you. And obviously there's two sets of questions since I have letters and numbers. So I can ask you quite a bit about those organelles. Now, there's a chart, a table in your textbook about the organelles and their functions. You might want to use that. Go in and look at the notes I gave you during class. I always take my test questions right off my chapter notes. My chapter notes I gave to you, right? I either talked about them or published them on the PowerPoints. Okay? All right, so mechanisms of membrane transport. There's not a diagram for that. So yes, we have diffusion. Yes, there's osmosis, right? There's facilitated diffusion, right? Then there's different kinds of transport, active and passive. All of these terms, do you know the difference, right? And it's not just, for example, this is the one I really like because it's so important to the cell. Glucose is water soluble. You know that because when you stir a sugar into water, you can't see it, it dissolves, right? So because it's water soluble, that means it can't go through the membrane, but your cell needs glucose to make ATP, a lot of it, right? So naturally, the cell would want to have a membrane transport mechanism that would help a lot of glucose come into the cell, right? But it doesn't make sense to bring glucose in to make ATP and use ATP to do that, right? So what is the mechanism that brings glucose into the cell? Insulin. Insulin is a chemical, but what's of all your... Active transport. It's actually facilitated. It's facilitated transport. That's where, well, you can check it out. The glucose molecule just has to dock with a little receptor, and the receptor opens and brings it in. No ATP required. Okay? So you'll have these questions about specific transport mechanisms, and then more general ones. Passive transport, no ATP needed. Active transport, you need ATP to move that material. And the active transport, an example would be your conduction of your electrical signals on the nerve membranes. Those are electrons moving down the surface, right? And when you do that, the electrons get trapped on the wrong side of the membrane after your action potential moves down the cell. So you actually have to pump back the ions that help you send that nerve signal back to the right side. You go in and look at that, right? You got to be a little bit more than term and definition. You should have an example that would apply to it so you can really understand how that's working. All right, intracellular attachments. That would be from this diagram right here. And there are basically these three kinds of attachments. Occluding means, well, it's the opposite of including. I don't know why they don't call it excluding. Right? There's no materials between the walls right there. They're like staples. A gap junction has these pores in the connectors. The pores are great for what?
Because we might need that space when this goes up.
Yeah, for moving things through, for transporting things through from cell to cell, okay? And I gave you an example about how the cells of the heart, they start a contraction of the atria in one part of the heart, but both atria contract at the same time. That's because the electrical signal through ions have moved through these channels through all the cells of the atria. That's one example. And then finally, these anchoring style connectors are like Molly bolts. They not only connect the cell walls, but they brace themselves with these plates on either side, and that makes these very strong for attaching the cytoskeleton. This would be along the wall of a cell. Here's down at the floor of the cell, the basal level. So I've translated that diagram into, are you ready? You guys are like the paparazzi. I feel like Angelina Jolie, except for every camera's away from me. Here you go, that's what it looks like on a test there. And again, don't just go, okay, B is these gap junctions. I may not even mention gap. I might say these junctions are particularly helpful in the heart for transmitting, the conduction for a heartbeat, right? So you got to be careful about studying, right? Get a more complete idea of how this all works. All right, so much for intracellular attachments. What's next on our list? The cell life cycle, which is including your interphase and your various phases of mitosis there. I think I mentioned, nope, we gotta mention that now. Is that you, Chris? Don't forget you have to take a makeup test. Did you miss one when you were being added? Okay. All right, so next, no. Where'd they go? Yep, there they are. Okay, so this is the diagram that's in your book. And your diagram nicely provides both an illustrated version and the histology version. I'm gathering that probably all of you, I didn't get one question about the whitefish blastula yesterday and Tuesday, right? Most of you in lab didn't look at those slides, okay? So the diagrams will be on the lecture test, but in lab you'll actually see mitotic figures, okay? So you're going to have to go back, and if you didn't see them in real life, you can go to 170. They have a microscopes and slides in there. They'll be happy to help you with that. Or you can check these out online and hope you can transfer these images to the ones that I use. Okay. I mentioned the other day in lab that I don't think my images are staying purple. So if that's going to throw you off, then you're going to have to get a more generalized idea. Okay? And you can always practice, go online and